Good day, everyone. I am Sherwin Zamudio from the College of Fisheries and Aquatic Sciences of Western Philippines University for the Princesa City campus. My study entitled The Survival of Boas Giant Clam Tridacnanoe Larvae, feed with two types of phytoplankton in Puerto Princesa City, Palawan, Philippines. For my introduction, first, what is Tridacnanoe? Well known as Noah's giant clam or teardrop giant clam, which belong to the phylum Mollusks in family Turdocninae, and it grows 6 to 20 centimeters length and has 5 to 7 radial ribs, and the color brown, yellow, blue, and green. And Turdocna novae, also confused with the Turdocna maxima, and first identified in Taiwan and Japan. And in Palawan, the first record of Turdocna novae was in Paraiso Resort in Puerto Princesa, Port Barton, San Vicente, Palawan. Why to conduct this study? First, various studies have been conducted about the other giant clam species, but not much on Tridacna novae. Second, no studies have been published yet on the survival of Noah's giant clam Tridacna novae larvae with two types of phytoplankton, specifically in Palawan. For my objectives, to determine the survival of larvae fed with two types of phytoplankton, and to determine the larvae size fed with two types of phytoplankton, Tetrasilmi species and Isochrysis galbana. The study was conducted at the hatchery of Western Philippines University, Binduyan Marine Research Station in Barangay Binduyan, Puerto Princesa City, Palawan. For the giant clam larvae, the larvae used for this study was collected from the Tridacnanoe breeders' stock of the hatchery, and the breeder were endorsed to a spoon using serotonin solution, and 2 days old larvae were used for the study, and the sample were drawn from a concentrated volume of larvae inside the plastic bin to determine the average number per ml. And 5 ml sample were drawn using a pipette, and the number of larvae was counted under the microscope using Sedgwick Raptor, and an average number of larvae per ml was used as the stacking basis in the experimental setup. For my experimental setup, 3 treatments were used in this study. In table 1, 9 beakers with a volume of 120 ml were set up in complete random design inside the laboratory. Each beaker was filled with 60 ml water containing larvae, or 1 larva per ml, to ensure a homogeneous distribution of larvae. The water was disturbed using a small homogenizer, or the balloon holder, before filling the beaker. Aeration was provided in each beaker with the use of air stone. For my phytoplankton feeding, two types of phytoplankton were used in this study, the Isochrysis galbana and Tetrasilmi species phytoplankton. New cultures of each phytoplankton were prepared 3 days before the start of the experiment, and the larvae were fed with 10,000 cells of each phytoplankton. And before each feeding, the number of phytoplankton cells was counted by drawing sample from the phytoplankton culture using a microscope and hemocytometer slide. The number of cells was determined by averaging the counts of 4 corner compartment of the hemocytometer multiplied by 10,000 to get the count per ml. Treatment 1 was fed with Tetrasilmi species, and treatment 2 with Isochrysis galbana, and treatment 3 with 50% of both the phytoplanktons. For my data gathering, the experiment was run for 6 days, and the sampling was done on the last day of the experiment. The larvae were collected pouring the contents of the beaker into 60 micron sieve, and the collected larvae were transferred in a small petri dish by squirting 10 ml of seawater in the underside of the sieve using a calibrated plastic pipette to collect the larvae. The petri dish was then tilted to concentrate the larvae on one side, and the number of surviving larvae was counted under the microscope using a Sedgwick rafter. The size of the larvae was determined using CPCE program, and the number of surviving larvae was recorded and samples of larvae examined under the microscope were photographed for the larval size. For the result, the survival of larvae with two types of phytoplankton, results of one-way ANUBA showed were no significant difference. However, the highest survival was treatment 2, which is the Isocrasis galbana, with a mean survival of 81.02%, and the lowest survival was treatment 1, which the Tetraselmis species. Larval size result of the one-way ANUBA on the larval length showed no significant difference among treatment, and the treatment 3, which mix, show a higher larval length compared to the two treatments with larval length. For the conclusion, no significant difference were noted in both survival and larval length within 8 days post-fertilization. Further, the amount of phytoplankton administered was sufficient to provide food to larvae in all treatments, and it served as additional information on the survival, feeding, and larval growth of Tridacna novae. Moreover, the larval stage at which the study was conducted could have also affected the result of this study. For recommendation, further studies on the larval growth and survival of Tridacna novae on extended time should be conducted to be able to obtain better results. Thank you for watching, everyone.